Hi, guys. Let's see, I'm trying to figure out how to work this. Let's see. Okay, hopefully you guys can hear me. And hopefully you can figure out how to ask questions. Can you see my screen right now? I don't really know what's going on. Oh, I am looking at Evan's screen. I don't want to do that. I don't want to do this. Okay. I don't know why it's sharing. Yes. Okay. Well, oh, I can see some people, kind of. Hi. I can't hear you guys though. Oh, I can unmute. Okay, now you're unmuted. Oh, and I can see you all.
Okay, let's put this up. Let's put it right here. Everyone, wait, no, put it right there. We can all stand behind it. No. What the heck? Oh, shit.
Okay, if I pull something up, can you guys see?
What?
Can you see my screen or no? Can you just see my face?
You. We can see you.
You can just see me?
We're the only ones in here. Okay.
No,
Okay.
these people are in here. Hey guys, please.
Oh god, I'm really close to the camera.
I'm like, oh.
Okay, so are we all at Starbucks?
Yes.
Yes.
We're having a nice little
Okay.
meeting at Starbucks.
Nice. Okay, let's see if I can unmute.
So everyone else is here. Oh, wait,
Okay.
Wait, I'm in this as well.
I'm unmuting everybody.
Oh, everyone's getting unmuted.
Oh,
Okay, so what questions do we have? And then hopefully I can figure out how to share my screen at some point.
Your question.
oh, we have a lot.
Personal note here.
What's the origin of a virus?
I'll let you.
What is the origin of a virus?
Yes.
Like, in what context? Oh, now my screen just says Nicole.
Wait, where should I go? Like, I'll just put it down here.
This is a funny suit.
Okay, can you still see me or no?
Yes.
To share your screen, all you need to do is click the share screen button that should be at the bottom of the
Yeah,
Really? Yeah, really? No, I made it up.
Why can't we see you?
okay, when I tried that before and I hit screen, then that's when it gave me Evan's screen.
It doesn't show you.
What about now?
Is it just you?
Oh no.
Oh, wow.
Do you see stuff now?
Yes.
Oh, okay. All right, so let me go.
You're not gonna echo. I have one over here. No, I can't hear her. Do it so you can see yourself. No surround sound. Yes. Don't be afraid.
So is that like a question from the study guide?
Yes.
The origin of viruses?
Did you like sign language or something?
So it's mostly just knowing that it's not really a cell, right? It's just
Matthew!
okay, I'm gonna mute you guys. Can I mute you? It's mostly just knowing that it's not really a cell, that it is just DNA or RNA in a protein code. So that's really all you need to know about the origin.
It's a DNA. What? I don't know. I can't hear over the
Comment something.
Wait,
Are you guys muting yourself? Should I mute you?
Probably. What you said? Should I mute you?
All right, next question. Who else has a question?
Another question.
Why you want to
Here, I'll type you to it.
Okay, if I mute you guys, yeah, just type in something.
What'd you say? She said.
I already did.
Okay, so I have a question of structure and function of a vesicle.
Someone get off this one.
So a vesicle is,
Could you explain that?
let me move this out of the way. So a vesicle is just a small, almost like a piece of the membrane that encircles something. So it can take things from the Golgi to the membrane and then fuse with the membrane and go out. So hopefully that helps with what a structure and function of a vesicle are. That is chapter. What?
I shared you with all the questions.
I didn't share this.
Describe the pathway of motion.
Okay, any other questions?
Wait,
Guys,
how do you?
I feel like I can still hear you guys, even though they're muted.
She can still hear us. I have my audio
I'm just listening.
really close to this. Wait, how did you
Okay.
do that?
So another question, compare and contrast what is in a prokaryotic cell versus a eukaryotic cell. So if we're looking at,
Oh, it's probably mine.
okay, if we're looking at if you guys are at Starbucks, can you like make sure you're muted? Because I only see Sam.
I don't know how to.
Is any, oh, there we go. Okay, that's better. All right, so let's see, compare and contrast what is a prokaryotic cell versus eukaryotic cell. So we have, here is a bacterial cell, right? A prokaryotic cell. So you can see there is the cell wall, cell membrane. We have DNA here and some ribosomes. And then when we're looking at a animal cell or a eukaryotic cell, we have all the organelles in addition to the cell membrane. So nucleus, ER, Golgi, same thing, plant cell, all the organelles, but plant cell also has the cell wall. So comparing them, right, they're both cells, they both have similar, some of the same structures, like the cell membrane, DNA, and ribosome. Okay, next one is describe the path of the protein from creation and how it's different if the protein is being exported or staying in the cell. All right, so that kind of goes with this. I'm gonna try to move this so it's out of the way. So if it is going to stay in the cell, right, a protein is made in the ribosomes. Ribosomes can be floating, or if they're on the ER, that is when kind of has to go through the full endomembrane system and get some processing and packaging before it leaves the cell. So let me see if I can find that slide somewhere here. Nope. Where is it? Maybe I missed that. Chapter. No, it should be in chapter 4. Okay, so we're looking at the picture where it shows the pathway. Here we go. So here is the rough ER. So proteins can be made in any of these ribosomes on the ER. Then that protein gets packaged into a vesicle, moves to the Golgi, right, where then it's modified, packaged, and then goes into another vesicle and then that vesicle will fuse with the cell membrane, and then that's where the protein is secreted outside of the cell. Now I lost my little, uh-oh. Where'd my little chat thing go? Oh man. Okay. No, that's not it. All right, I'm still getting used to this. There, okay. Okay, compare and contrast cell junctions. So I do have that in here. Can you guys see if I just have this screen like this? Is that okay? Yeah? No? Okay, thank you. I can't hear anybody, so it's weird. Okay, so let me go to the cell junctions. Here we go. So plasmodesmata are in the plants. So this is where water and small solutes can go through. That is similar to in animal cells, the gap junctions. In animal cells, there's also tight junction and desmosomes. So the tight junctions are really tightly packed together. Desmosomes are almost like a rivet. And then gap junctions, there is a gap. So this is what it looks like. Tight junctions really close together. The desmosome is like a rivet, and then gap junctions, things can get through. Ooh, what are important key parts of chapter 5? All of it. I can't really give a quick summary. But if you have more specific questions, I can even narrow it down a little bit more from like 5.3 if you can. So like more specific, the better. I know, I'm sorry, I realized after that you're missing the Bears game. All right, it's only 40 minutes though, you can still see part of it. Explain the relationship between membrane potential and electrochemical gradient. Okay, so membrane potential is sometimes, and we'll get into this more when we talk about the nervous system. Let me try to find that slide. That was in, okay, here. So this slide. So a lot of times what happens is there's a membrane potential or there is a difference in ions from one side of the cell to the other. So in this picture, you can see that the concentration of hydrogen ions is higher on the outside of the cell than in the inside of the cell. So typically, where are the hydrogens going to go? They're going to go this way, right? You would think that they would go down the concentration gradient through, it would have to go through a channel, right? It's an ion, so it can't just diffuse in. So, but here we have a proton pump. So what's happening is this is active transport, right? And it's moving against the concentration gradient. So hydrogen is actually being pumped across using ATP against the concentration gradient. So that is keeping that electrochemical gradient. So that's kind of addressing that question. You guys can turn your audio on too if it's easier for you to ask a question. It's just hard when you're in a big group to hear. But if you would rather ask instead of type, that's fine too. Well, you have to know water potential on the test, yes. So you will have to calculate water potential and understand what it means and what that means as far as where is water going to go. So we can review that sheet tomorrow in class. Did you guys finish that today and check your answers? Anybody?
I did.
Okay, thanks.
Same, Evan.
So we can go over that a little bit more if you guys feel like you will need it. But yes, you will have to calculate water potential and know what that means. Other questions? Okay, G protein is just one of those ways that cell signaling can occur. So a G protein is part of the cell signaling pathway, so it's part of reception here. So a G protein could be involved kind of in this reception process. So if you see, yeah, in this slide, so this G protein here, when it's activated, slides over and changes the enzyme shape, which then starts the signaling cascade or kind of the transduction part. So it's just one of the ways that the signal cascade can start in a cell or on the plasma membrane. So G protein activated by GTP, when it's activated, this is the G protein here, it slides over and then activates this enzyme, and then that's where then transduction happens down here. Explain the transduction by cascades of molecular interactions. Benefits, role of protein kinases and protein phosphatases. Okay, so that's kind of going right along here. So this is just the other type of reception where we can have, this is just the ion, the ligand-gated ion channel. So this is where that signal can come in, bind, which then makes this shape change. So you'll see a lot of cell signaling is changes in shape because a lot of this is all proteins, and proteins are very shape specific. Shape determines function. So here this gate is closed. When this ligand binds, it actually changes the shape of the protein, which allows this membrane protein to open. And then if we get into the signaling, then here we go. So here's the signaling molecule. Let me go back to your question to make sure. All right, benefits, role of protein kinases and protein phosphatases. Okay, so here we have an inactive protein. So what happens is it can be activated by, okay, this is really bugging me. Oh, maybe it'll work now, maybe it'll stay up. Okay. So it can be activated by a protein kinase. The kinase is the enzyme that will take this ATP and break the bond, releasing a phosphate, and then that activates the protein. So the kinases are responsible for this chain, this kind of transduction to occur and for the signal to go through the cell. So kinases basically put a phosphate on a protein, which activates it so it can keep transferring the signal. I'm trying to find the comment again. Sorry. Okay, the protein phosphatases are what then takes the protein off of here, right? Because if this, phosphate, not protein, here's the protein, here's the phosphate. If this phosphate stays on, the signal keeps going and going and going, and the cell doesn't want that to happen. So we need the phosphatases to come along and take off these phosphates so that the proteins can be inactive again and then can be reused the next time the cell needs it. But we don't want it to stay on forever because then that would keep causing this cellular response to occur. And the benefits again are just that the signal is amplified and also to get the signal through the cell. From the cell membrane, it has to go through and get to the nucleus. So you can kind of think of again of the dominoes, you know, if we start in one spot and we need to get to another spot, we need something connecting that activation to that actual specific cellular response, which a lot of times is in the nucleus with a segment of DNA, that gene being transcribed into mRNA and then eventually making a protein, whatever protein that cell may need. Structure and function of vacuoles. So vacuoles are basically some sort of storage. You can think of a vacuole like a storage. So there's a big central vacuole in plant cells, right? Plants need a lot of water, so typically that will hold a lot of water. Animal cells do have vacuoles as well. They tend to be smaller. They can also hold food, water, or waste. So that is basically their structure and function. It's basically just a big storage compartment. All right, explain transduction by cascades of molecular interactions, benefits. Okay, I think I just did that. Hopefully, Haley, that answered your question too. And Kale and So can I differentiate between isotonic, hypertonic, and hypotonic and how this will affect osmosis? Okay, let me see if I can go back here. I know I have it. Oh, here we go. Okay, so really quickly, let's just do passive and active transport. So this, I'll put this over here for a second. Okay, so this diagram is showing passive transport, right? So we have diffusion, which is a form of passive transport, right? We're moving from high to low concentration or along the concentration gradient, and small nonpolar things, right, can travel through the cell membrane into the inside of the cell or out if that's the way it's going. Facilitated diffusion, right, same thing, no energy is required. We're moving from high to low concentration, either through a protein channel, or this is kind of the other mode of facilitated diffusion, where it looks like there's a shape change going on. This is a carrier, right, but there's still no energy required. We're still going from high to low concentration. So these kinds of things, glucose, right, can use facilitated diffusion. Water, right, uses the aquaporins, which are specific to water. Over here, active transport, we are going from, could be either from, can you guys see this? Or is this in the way? Do you need me to move this? All right, we'll move it over there. Okay, so active transport, you can see that sometimes things are moving opposite. So it could be moving from low concentration to high, and that's why it needs to be pumped across. This is showing the protein pump. So going from low to high concentration, so against the concentration gradient, we would need that ATP to move it across. Exo and endocytosis are also forms of active transport. Because molecules are so big, they need to be surrounded by a vacuole and either pushed in or out of the cell. So that's kind of the two main types of transport. And then once we get to osmosis, when we're looking at that, so if we're looking at this U-tube, if we have a sugar molecule here that cannot cross, this is like a selectively permeable membrane. So this is kind of like another way to look at a cell. So we have lower concentration of solute over here, higher concentration over here. So we know if this can't move, water is going to have to move this way, right? So water is moving this way to where there's a higher concentration of solute. Okay, it's still moving down the concentration gradient if we're talking about water, but remember it's opposite when we're talking about solute. So wherever there's more solute, that's the direction water is going to go. As far as hypotonic, isotonic, hypertonic, here we have hypotonic. In a hypotonic environment, an animal cell is actually going to lyse, or it's going to explode, because your blood is not pure water, right? We have all kinds of electrolytes. One of them is salt. So that's why if you've ever needed an IV fluids is actually saline. So it's usually like 0.7% salt, because that's what your blood is. So animal cells would lyse in a hypotonic solution. So if it's pure water, the cells will explode. Isotonic solution, right? Water is always moving. So don't just think water is moving in or water is moving out. We do have that back and forth, okay, of water moving in and out. So isotonic, it's just that the net flow is zero in an animal cell. Hypertonic, if the solution has more solute than what's inside the cell, right, water is going to leave, a net loss of water from inside the cell, and the cell's going to shrivel. It's a little bit different in plant cells, right? In a hypotonic environment is the normal environment for plant cells, and they're going to be turgid. So this is why they spray plants like at Jewel or any grocery store. That's why they spray them with water, so they look nice and healthy. Because the cells then, because they have that cell wall, they won't lyse. So that's in a hypotonic environment. Isotonic, they're still okay, but they're just not as full of water. So they might look a little wilted. And then same thing happens. If plant cells are in a hypertonic environment, they shrivel because water is leaving. So always think about where there's going to be more solute. So wherever there's more solute, that's where the water is going to go. So that's either going to cause the cell to expand or shrink, depending on that water flow. Okay. All right, well, we need to understand the parts of the cytoskeleton. I don't necessarily think you need to know all the different parts. You probably should know that myofilaments are involved in movement, and that just the cytoskeleton in general is what builds up cilia and flagella. So again, that movement piece is important for cytoskeleton. And then just know that it's, kind of the basic structure of the cell. So kind of keeps things all together. Okay, so we already talked about vacuoles, membrane proteins, channel proteins versus carrier proteins. Hopefully you saw that. So the channel proteins look like, oh yeah, I just described that. So this would be the channel protein, right? This would be the carrier protein. So very similar. It's just kind of that they are both, you really don't need to know a lot about how they're different. Just both of these are involved in facilitated diffusion. So both of these are involved in passive transport. It's the protein pumps that are active transport. And the way I remember that is like if you're pumping something, right, you have to expend energy. It takes energy to pump. So that's a form of active transport. Differentiate between cell signaling used by neurotransmitters and hormones. So a lot of times neurotransmitters are going to use the cell signaling where it binds on the membrane. Oh, wow, we already, okay. It's just told me that we removed the 40-minute time limit on your first group meeting. Okay, it's like we haven't been talking for that long. So anyway, so the neurotransmitters typically have to bind to a membrane receptor right here. That is typical for neurotransmitters. Hormones, remember, are typically lipid-based, so they can travel through the cell membrane and go into the cell, and then they will probably have an intracellular receptor, so they don't have to bind. The ligand does not have to bind to an extracellular or membrane protein. It can actually travel through the membrane and start that way. Let me see if I don't think I put that on there. Other questions? We have to calculate the volume and surface of a cell. Yes, you may have to do that. So those equations are on your equation sheet, and you will have that for the test. But you won't ever have to memorize formulas or know how to do it. It's just that you have to plug in numbers that are given to you. But you always have your equation sheet on the test. This is really weird. I don't see anybody's face and I don't hear anyone. Any other questions?
Med Club meeting tomorrow.
Yes, Nicole, Med Club meeting tomorrow. I'm trying to think of anything else we have not gone through. Everybody okay on viruses, the lytic versus lysogenic cycle? Anything else about cell transport that we need to go through? Okay, so ways to stop viruses. So there's prevention, right? So that's where vaccines come in. So we can vaccinate against viruses. There are some antiviral drugs that help, but really there's not a way to stop a virus. Oh, Derek, they're hidden from you. I'm looking through my slides to see if there's anything else we need to talk about. These are like the main things I kind of put together this presentation to see, or what I thought would be common things that we would need to go over. So I think we basically covered everything. Anybody have any other? Nothing else? We're ready to end? Okay. Well, that is it. Thanks for joining me, and I will try my best to upload this. And if you think of anything else, or you want to make sure we cover anything tomorrow in class, let me know. Make sure study guys done all that other stuff we talked about today. And I'll see you guys tomorrow. Bye.